Hello everyone. Today, let us discuss on the topic factors affecting ripening. Let us start with the introduction. Ripening is the final phase in the development of the fruit. It is the process by which the fruits attain the desirable flavor, quality, color, palatable nature and other textural properties. In general, a fruit becomes sweeter, less green, and softer as it ripens. A lot of factors affect the ripening process both under pre-harvest and post-harvest conditions. Climatic fruits like mango, banana, papaya, sapota, and custard apple are often harvested in a mature but unripe condition and then subsequently allowed to ripen by natural release of ripening hormone, that is, ethylene, from the fruit. However, natural ripening in some fruits is a slow process, which leads to high weight loss, desiccation of fruits, and uneven ripening. With the rapid development of fruit trade, artificial ripening has become essential. A further advantage of controlled ripening is to improve uniformity of ripening of fruits. So let us now discuss on some of the factors that affects the ripening of fruits. Temperature. Most fruits ripen satisfactorily over a wide range of temperatures, but unsatisfactorily at temperatures beyond this range. Below a critical temperature, many tropical and subtropical fruits develop a disorder known as chilling injury, which inhibits ripening. In banana, this critical temperature is between 10 and 13 degrees Celsius, while in Koch's orange pippin apple, it is as low as 3 degrees Celsius. Many fruits fail to ripen beyond these 30 to 35 degrees Celsius. The inhibition of ethylene biosynthesis, respiration, and color changes due to higher temperature are reversible within a relatively short period after return to the optimum temperature. The relationship between ripening period and temperature is due to fruit respiration. Fruit respiration depends on many enzymatic reactions, and the rate of this reaction increases exponentially with increase in temperature. Water loss or humidity. Relative humidity is a term used for expressing moisture content in air. Relative humidity, it is defined as the ratio between the vapor pressure of water in the air to the saturation vapor pressure possible at the same temperature, and it is expressed in percentage. There is a fairly good relationship between the degree of water loss from the fruit with the relative humidity of air. At an ambient temperature, relative humidity of 95 to 100%, fruit loses little or no moisture, and ripening period is unaffected. However, as humidity decreases, the rate of water loss increases and ripening period reduces. Fruit ripen better with not only better appearance because of the absence of shriveling, but also with better internal quality at a relative humidity of at least 90%. High relative humidity, that is more than 95%, can encourage the growth of bacteria and fungi. It has been found that 90% relative humidity is usually the best compromise condition for fruit storage. The humidity of the post-harvest storage environment also has an effect on the rate of ethylene production, thus affecting fruit softening. Persimmon fruit kept under conditions of high humidity produce little detectable ethylene, whereas fruits kept at low humidity produce significant amount of ethylene, thus accelerating fruit softening. Fruit shading. Exposure to direct sunlight reduces the ripening period of fruits. Sunlight increases fruit temperature above ambient temperature, which increases respiration and possibly the rate of water loss. The amount of increase in temperature depends on the intensity of the radiation, the size and shape of the fruit, and the duration of exposure to direct rays of the sun. The intensity of solar radiation depends upon latitude, altitude, season of the year, time of the day, and degree of cloud cover. Maturity of the fruit. Immature fruits may ripen, but typically will not have full flavor development and may not have full cosmetic appeal. Ripening such fruit will require extended ethylene application and a longer ripening cycle. There is a direct relationship between maturity, time to ripen, and fruit quality. Earlier the harvest, longer is the time to ripen. Greater the maturity, lesser are the number of days required for the fruit to ripen. Ripe fruits from early harvest had very poor quality, indicated by the lower organoleptic ratings, and with increasing maturity, quality improved. The ethylene factor. The commencement of natural ripening in climatic fruit is accompanied by an increase in ethylene production. Treatment of pre-climatic fruits with exogenous ethylene advances the onset of ripening. This response is used widely in commercial practice to achieve controlled ripening of fruits such as the banana. Ethylene produced by other fruits or from outside sources may accumulate in a storage chamber containing unripe fruits to a level that initiates unwanted ripening. Often during marketing, several types of fruits and vegetables are stored together, and under these conditions, ethylene given off by one commodity can adversely affect the another. Coal gas, petroleum gas, and exhaust gas from internal combustion engines contain ethylene, and contamination of stored produce by these gases may introduce sufficient ethylene to initiate ripening in fruit and promote senescence in leafy tissues. In addition to methods which reduce the ethylene concentration, the sensitivity of the produce to ethylene may also be lessened by storage at low temperature and by either raising the levels of carbon dioxide or decreasing the level of oxygen. In practice, ripening or senescence is delayed by maintaining ethylene at low levels in storage rooms. This can be achieved by storing ripe and unripe produce in separate rooms and by ensuring that gas pipes, gas cylinders, and exhaust gases from internal combustion engines are kept well away from the storage rooms. Oxygen and carbon dioxide concentration. Normally, fruit respire and ripens in air which contains by volume 78% nitrogen, 21% oxygen, and 0.03% carbon dioxide and water vapor and inert gases. Modifying the atmosphere can improve the shelf life and reduce wastage of certain foods. High oxygen levels in the post-harvest environment of climatic fruits have shown to enhance fruit ripening by increasing ethylene production, resulting in accelerated fruit softening. Raising the level of carbon dioxide and decreasing that of oxygen in storage atmosphere delays ripening. Similarly, addition or removal of carbon dioxide and ethylene also regulate ripening. Low levels of oxygen, that is 1 to 5%, prevented ripening of banana as indicated by the onset of endogenous ethylene production, the respiration climacteric, and the change in color index. Oxygen tension greater than 1 atmosphere tend to promote ethylene biosynthesis, consequently promoting ripening. Higher carbon dioxide slows ripening by preventing ethylene to accelerate ripening even at higher concentrations, since carbon dioxide acts as a competitive inhibitor for ethylene action. Increasing the concentration of carbon dioxide to about 3 to 10% in storage atmosphere delays the onset of climacteric respiration. Mechanical damage. Mechanical damage is a physical factor affecting ripening. Fruits respond at the physical and physiological levels to mechanical damage. Physical responses are mainly linked to the appearance with the source of symptoms of advanced lignification and necrosis in the damaged region. Due to the physiological responses, mechanical injuries cause alteration in color and flavor, tissue softening, and faster ripening. These are mainly due to the increase in respiration and ethylene production. Increased weight loss, increased invasion of microorganisms, and higher enzyme activity in the affected area. If ethylene production is sufficient to start the climacteric respiratory response, fruit immediately starts to ripen. Damage can also reduce ripening period by causing moisture loss. The effect of damage can easily be measured by recording fruit weight loss over time. Cuts and abrasions on the surface membrane cause the most weight loss. Altitude. Within a given latitude, the prevailing temperature is dependent upon the elevation when other factors are equal. There is on the average a drop in temperature of 6.5 degrees Celsius for every kilometer increase in elevation above sea level. Storing food at high altitude will therefore tend to increase the storage life and decrease the loss in food, provided it is kept out of the direct rays of the sun. Air movement. Air movement over the produce is a significant factor influencing the rate of moisture loss. Air movement is required to remove heat from the produce, but its effect on moisture loss must also be considered. The faster the air moves across the surface of the fruits and vegetables, the greater is the rate of water loss from the produce. Greater is the rate of water loss, greater is the rate of ripening. Thus, the rate of ripening of fruit is hastened with an increase in air movement in storage rooms. Biotic stress. Fungi, bacteria, viruses, and insects also account for considerable proportions of total post-harvest losses. Pests and diseases reduce both ripening period and the overall quality. Treatment of the fruits. Other than the above factors, fruits can also be treated to increase or decrease ripening. So, let us now discuss on some of these treatments. First is washing with ozonized water. Ozone is the most effective natural bactericide of all the disinfecting agents. Ozone is the safe and natural purification and disinfecting agent. Treating commodities with ozone water not only killed the adhering microorganisms but also neutralized the ethylene gases produced by ripening or decay. The reduction of ethylene gas helps to preserve reduce shrinkage, and increase the shelf life of fruits. Then, the next is calcium application. The post-harvest application of calcium chloride or calcium nitrate play an important role in storage of fruits by maintaining their firmness and quality. Treatment with 1-methylcyclopropene. 1-methylcyclopropene is a synthetic cyclic olefin that inhibits ethylene by binding irreversibly with ethylene receptors. The affinity of 1-methylcyclopropene for the receptor is approximately 10 times greater than that of ethylene. The effect of 1-methylcyclopropene on delaying ripening and senescence rate are associated with the reduced ethylene production, respiration, color change, and rate of softening. Polyamines. Polyamines are low molecular weight and small aliphatic amines that are ubiquitous in living organisms and have been implicated in a wide range of biological processes including plant growth, development, and response to stress. The polyamines inhibit biosynthesis of ethylene and thus delay ripening. Polyamines also maintain firmness of the fruit due to the cross-linkage in the carboxyl group of the pectic substance in the cell wall, resulting in rigidification, thus blocks the access of degrading enzymes and reducing the rate of softening and less prone to mechanical damage. Ionizing radiation. Ionizing radiation causes delayed ripening, destruction of microbes, minimal insect infestation, and changes in several biochemical processes. Generally, low radiation doses is applied to fresh fruits. Gamma rays in preclimacteric fruits reduce not only ethylene production but also the sensitivity to it. In cases where irradiation delayed ripening, the fruits subsequently either fail to ripen or exhibited an anomalous ripening, and were of poor quality except in bananas, subjected to low doses. In many fruits, the degree of maturity may be critical with regard to the feasibility of irradiation as a treatment to controlled ripening. Other chemical regulators. Other than ethylene, abscisic acid is also an important promoter of ripening. Concentration of abscisic acid commonly rises late during fruit development or ripening. Abscisic acid stimulates ethylene production in preclimacteric fruits. Auxin, cytokinin, and gibberellic acid invariably have an effect opposite to that of abscisic acid. Endogenous auxin acts as ripening inhibitor and modify the tissue response to ethylene. The role of GA as a senescence delaying agent has been demonstrated in fruits. It interferes with the degreening of chlorophyll as well as with the biosynthesis of carotenoids and anthocyanins. Cytokinins also interferes preferentially with certain senescence phenomena, mainly delaying color changes, and is associated with maintenance of the endogenous GA level and prevention in increase in ABA-like compounds. Now, coming to the conclusion. Fruits are generally harvested when fully mature and are ready to begin ripening. Even after harvest, the produce is still living as it continues to perform the metabolic reactions and maintain the physiological systems which were present when it was attached to the plants. The process of ripening can be controlled within limits by temperature regulation, altering external atmosphere of oxygen and carbon dioxide, and subjecting the commodities to different treatments. This must, however, be achieved keeping the deleterious effects such as water loss, microbial infection, and development of physiological disorders to the minimum. These treatments have implications on the post-harvest life of the fresh fruits.